So I'm calling this Diverse Estuaries in Your Backyard, and I also want to start off by acknowledging everybody. I've got most of my PIs and graduate students are up there. There are a lot of different people on here, multiple universities. We work with the Weot Tribe and H.T. Harvey, so it was a really pleasant collaboration. And in continuing those acknowledgments, thank you for Ocean Protection Council for funding us, and Sea Grant for running the money, and Fish and Wildlife for taking care of us and overseeing all the project. There were a lot of HSU students out here supporting our work and volunteering for all the muddy sessions and doing a lot of kayaking. And a lot of these people on the right-hand side of my slide up here were helping us with permits and access and a lot of issues. Took a major effort to make all this work happen. Okay, estuaries. Recognized as being amongst the most productive ecosystems on the planet. They're right up there with coral reefs and tropical rainforests. They're also recognized as being amongst the most threatened ecosystems. And it's not a surprise, right? Because they're right on our coast, and so the first places people settle on the coast are the shores of the ecosystem and the watershed leading into the ecosystem. So on the conservation literature, when you're reading about the estuaries, they're going to tout all the ecosystem services they do. They're also going to tout the fact that they're extremely threatened. And we care about the estuaries because in addition to their productivity, there's just more and more services continue to be identified by what's happening in the estuaries. They have nursery and refuge functions. They have water clarity functions. They now have carbon sequestration functions. Eelgrass can sequester carbon in its rhizomes under the mud at the same rate that terrestrial forestry, temperate forest trees do. And now some of the plants and macrophytes in there may have a buffering capacity and prevent and buffer OA events as they come into the bay. So it's really important what's happening in there in the ecosystems. The same conservation literature points out, though, that conservation efforts have been they're either terrestrial focused or they're marine focused. And actually the fourth stepchild of conservation are the estuaries, even though they're so important and so threatened. So, we appreciate the fact that we have marine protected areas at all. There are 22 estuary marine protected areas in the state of California. There are 4 in our North Coast region from Point Arena north. The project that I led up here is the first one in the entire state to do baseline monitoring of any estuary. So the estuaries that we picked up here, I'm going to give you paint a picture for more of them in a moment, but we did the Mad River Estuary, which is not an MPA, and then we did Humboldt Bay and a couple others south of Cape Mendocino. And there's good news here and maybe some not good news. It kind of depends who you are in the audience. When it comes to the creation of these estuary and MPAs, you can do in those estuary MPAs today what you could do before. The creation of the MPAs did not stop any of the take activities to speak of. All right, so there is not an expectation of an MPA effect for these particular estuaries, which makes them different from a lot of the other MPAs and other habitats. However, there's 2 real points I would tout for their value. One is that, for specific species that the agencies care about, we're getting baseline information on that, for some of the commercially important fish and invertebrates. And secondly is that the estuaries that we're providing this information for are very poorly known. I don't think Katie touted this fast enough, but basically the Mad River Estuary has never been described. And a lot of the other estuaries that we're going to include up here are also very poorly described. So the information we're providing about that is important, and I would hope that information gets out and raises awareness, and that may be more important than enforcing no take. We'll see. So here are the MPAs up here, and in the pink are the 4 estuary MPAs in the North Coast region. You see the 3 of them south of Cape Mendocino up there, and you see the Humboldt Bay up there, of course. Now, this is also a context up here. So in yellow, you have all the other possible estuaries we have on this coast. So it's great that we have estuary MPAs, but there's a lot of estuary systems up here that are not included in the MPA system. The objectives of our baseline study were to describe the estuarine biodiversity, to provide more detailed information on a specific target species, and as best as possible to come up with a physical context information for each of those estuaries, and then to come up with recommendations. And so for my talk today, I'm just going to focus on, just introduce you to some of these results. So starting with the Mad River, going north to south up there, we picked the Mad River again for the reasons Katie said. I was kind of looking for a small linear riverine estuarine system north of Cape Mendocino that we could tractably sample, and that was going to be easy for us to get to, and that was definitely the Mad River. And then there's Humboldt Bay in the southwest corner of southern Humboldt Bay is where that MPA is located. 10 miles north of Fort Bragg is the 10-mile river MPA, and right on the south side of Mendocino town is the Big River Estuarine MPA. Gorgeous places. In terms of our study design and sampling, as I indicated, there is not an expectation of an MPA effect right now. So you've heard several other talks today where you had a reference site type of design. That didn't make sense for us, so we didn't try to do that. What made more sense for us was to try to pick representative estuaries, track them through time, and then, if there's a change in management action in an estuary, or if there's just some event in an estuary, whether that's natural or anthropogenic, then we could do a before-after comparison, and we'd have the data that would allow that to happen. And so that was our rationale for our approach. For each of the estuaries up there, we sampled 2 sites. So if it was an MPA, we had to keep our sampling within the MPA boundaries. We had an upriver site and we had a mouth site. Within each of those 2 sites, we had transect and traps in the mid-intertidal mudflats and a low transect. It was either very low intertidal or shallow subtidal. For each of those locations, we did biodiversity. So we did fish, did a lot of the mobile invertebrates in the infauna, algae, and seagrasses. And then, as you can see, we targeted certain specific organisms in each of those categories up there. We started in June 2014. We finished in June 2016. Just a little bit about the sampling in there, where we've got quadrat transect techniques for all the things that would require percent cover, like the algae, the macrophytes. Infaunal hole size classes is how we were trying to getting at who was down in the ground in there. And you see us using some bizarre techniques there to not disturb the substratum, us hanging over the bow of the kayaks there, which, yes, did provide a constant source of amusement. So sampling in the infauna, sieving that, using a variety of different types of traps up there to capture mobile invertebrates of various kinds, and the fish trap in the lower right-hand corner going for the larger crabs. And the beach seine, as Katie described. So each of those sites got 3 seines normally there. We're done. See a couple examples right there, and they're processing the fish, identifying them and sizing them and recording that information, and all those fish were returned alive after they were processed on site. Okay, probably the biggest thing that blew me away in being part of this project, because my history is rocky intertidal, a lot of time looking at eelgrass in Humboldt Bay, then getting onto this project, and wow, are these estuaries really different from one another. In hindsight, it is a really good thing we did not try to go with a reference site type of design. That would have been a bit of a train wreck, because the estuaries are so physically different from one another, they wouldn't have been good comparisons. A lot of the MPA studies all grapple with this. I know, I think it's a really extreme problem, would have been for the estuaries. But it's also really cool, and I hope I can impress that upon you. I think we all understand Humboldt Bay, some people don't even want to call it an estuary. When you look at the larger literature, though, there's wide recognition. There's a big gradient of estuarine types. So Humboldt Bay is extremely oceanic. Mad River, mostly open. It can close down a little bit in the late summer, but it's mostly open to the ocean. What makes Mad River also different, though, is it's got a dam, right? So when we have these riverine estuaries, it's not normal to have summer freshwater flows coming down there. The summer flows should be going down. that saltwater wedge should be getting further up. If you have a dam releasing fresh water, you change the hydrology of the system. So the Mad River in that way is not a normal system. Big River, totally open mouth. Big River estuary is 8 miles long inland, and it's a totally open. It's a really beautiful area. 10-mile river is, I'll just call it a seasonal lagoon because it has what we call a perched beach. And what's going to be what happens here is the beach builds up as the summer progresses and it becomes a dam. And I just got data from John Largier, who's our physical oceanographer at Bodega, and no one in my group has seen this, so surprise. Here we go, all right? So what you're looking at up here in blue is water depth. It's not referenced to a tidal dam, it doesn't matter. What you're looking at is the tide coming up and down, it's starting here in about August 1st, 2014. In the 10-mile river, the height of the low tide is getting higher and higher and higher, and the reason what's happening right there is the beach is getting higher and higher and higher, so the ocean is getting less and less and less able to get into the lagoon. Then we hit this point by about here, and we stop being tidal at all. The beach keeps building because of what happens to our beaches during the summer. The water starts piling up behind the beach. That's the water coming out of the watershed. Then in a matter of a few hours, here sometime in early November, too much, it just was too much, and the beach lets go, and it goes on down and crashes on down. In the meantime, we get a lagoon going on here. Water temperatures going up, and other data from John that he just sent me. Dissolved oxygen is going down, the lagoon is getting stratified, dissolved oxygen is going down. So there's this estuary that oceanographically should be situated to deal with wonderful upwelling, but it's not getting it because there's a dam in the way, and it's getting hot, and it's losing oxygen. So it's a really stressful place for animals, and across the context of a year, it's super variable. There is the concept of mean low or low water doesn't really exist in this estuary. Okay, so I just really want to impress upon you how different that is. Here's the mouth, probably of the 10-mile river built up as high as it's going to go, and that's a shot from this September. And then when it's broken out, that's the change. That's a pretty impressive change right there. Whoops, that was bad. Okay. Okay, recovery. All right. So, kind of ecological point, right? We have these gradients out there. Let's think about what the effect that I just told you is going to have on biodiversity. We've got gradients going north to south with potentially more upwelling below south of Cape Mendocino. We have these changes over time. We have the blob in 2014. We have El Nino in 2015, 16. So we have big changes with time. And then the particular estuaries all have their own unique characteristics. When you put that together, you realize you have a lot of different combinations of physical attributes, which ought to give us potentially going to give us a lot of different types of communities. And yeah, I know. Just focus on those nice circles. Okay, so these are also standard deviation ellipses like Katie was using right here. This is just for the macrophytes, so just for the seagrasses and the algae. And if I just walk this through with you, super oceanic, the two sites, there are 8 ellipses up there because there are 4 estuaries and 2 sites in each estuary. Okay, so Humboldt Bay, super oceanic, next most oceanic, both sites in Big River, next most oceanic or a little less saline, the 2 sites in 10 Mile. Then the mouth side at Mad River is close to that, and the upriver site at Mad River with the lowest salinities is pulling away from that. All you need to get out from this is that they're separating out a lot in space, and if I put up the fish ordination and I put up the invert ordination, they would be the exact same result, with the exact same take-home message that there's a lot of variation in biodiversity spatially. In other words, there's a high beta diversity. Go to a new site within the same region and you get a new biota. So, meet your Humboldt Bay MPA neighbors. I'd like to at least put a face on some of the reason why the ordination was different. If you can look at some of those organisms, This was certainly one of the fun parts of it, but I'm not cherry picking these images. These are the things that were driving the differences in that ordination. I'll just show you a couple slides just from Humboldt Bay and Mad River, since we're on Humboldt Bay and almost on Mad River. We all know about the eelgrass, but there's a lot of unique seaweeds up there, and quite a lot of the invertebrates that we'd expect to see with an eelgrass community, as well as a lot of the fish in here. Staghorn, tube snout, crangon, shrimp, pipefish, top smelt, surf smelt, herring, goby. How did I do? Anchovy. Oops. So close. I memorized that. And we go to Mad River, and we don't have eelgrass in Mad River, but we have another seagrass. We have rupia in there. I didn't even know we had it locally. It's a big brackish indicator. We have Garelia, that is not ulva. So we have some really unique algae, then we have some really neat invertebrates. These are a lot of the things that Katie was just talking about, and the gut contents of our fish there. And we even have some of the same pictures of the salmonids up there as well. Everybody wants to know, we're talking about the unique oceanic conditions that occurred during our study right here. And what did we find? Well, at the biodiversity level, I have 3 plots, one for the macrophytes, one for the invertebrates, and one for the fish, and they all look like this. At the community level, there wasn't a change, okay? We didn't see that big change, right? Which is interesting, but that's the whole community level. But for some of the target species, all right, we focused on some of those, we saw big changes. And yeah, there's lots of data and lots of analysis behind this, but I thought pictures were good. So in terms of multiple measures of abundance, eelgrass cover at both Humboldt Bay and in Big River, the 2 estuaries which had the most eelgrass, was quite high in summer, or relatively speaking, was highest in summer 2014, and it's stayed down in summer 2015 and 16. Staghorn sculpin, which is a fish I've really grown to like. Not handle so much, but from a data perspective, I like staghorn sculpin because they were present in every single estuary, in every single site. You can sample them, and if there's a signal in the environment that's going on, you can actually say something with confidence. And you see the exact opposite trend in the staghorn sculpins in here. When I was talking to Tim earlier today, that's probably a recruitment issue going on there. But I mean, we can't tell just by looking at the abundance numbers, but so there's some phenomena that was going on to explain that. So at a biodiversity level, not a change, but for specific target species, definitely. So to summarize, large spatial differences in biodiversity, no annual differences in biodiversity, but examples like the eelgrass and staghorn sculpins did change. And just as a context comment, so our sampling that we did probably doesn't even describe the biodiversity that exists in the MPAs that we visited, all right? Because we're limited on the number of sites we could do. That's one scale issue. Second scale issue is that the marine protected areas often don't represent even the estuary in which they occur. In this picture up here, this is the Big River here, and this is where in blue is the MPA. Well, many miles, the estuary goes many miles up the river. And the third large-scale issue is that the MPAs themselves are missing some of our most giant riverine ecosystems. So in a study like what we just did, we just scratched the surface of what's going on for estuarine diversity. Thank you very much.
About 2 minutes.
2 minutes for questions?
One observation in the North Coast is most of the lagoons, most of the rivers block for periods of time, and the salmon then are located more and more concentrated near the mouth because they're waiting to get up or into the lagoons. Do you think that's an area that needs further study?
I would put that to our fish people in the estuary. I don't know what our fish data were for like I'm trying to think what our specific fish data were for our systems.
I can partially address that. So our beach seine, in my call-outs to recruit volunteers for my monthly sampling, I would say, who wants to use a really big net to catch really little fish? Because it's a 150-foot net with a quarter-inch mesh. So what we're catching is really the juveniles. We did catch some steelhead, trout-sized steelhead, but we're in the river, and we're catching the juvenile fish. We're catching them as they're leaving the estuary. So that blockage that you're talking about as they're moving in, our data wouldn't have anything to say.
'Cause I know the Mad River got blocked in the 1860 or 80, and people went out and clubbed and speared salmon on the ocean side, and the run has never recovered even to this day from it. And now that was an extreme situation, and of course the Mad River has dams, and the Mad River's been logged and stuff, but I've always wondered whether that's a key part of the salmon life cycle on the coast.
I don't know, but we'll field questions on this later, happily.